Yes, I was putting my talk title together, and my Spotify Wrapped came out and really judged me a bit, so I felt the need to honor it. Yes, we study, I'm from Elke Mühlberger's lab, and we study a lot of filoviruses. I think probably everyone here is at least a little bit familiar with filoviruses, at least Ebola and Marburg viruses. And some of the others that are generally associated with these cute little guys, the Rosetus aegypticus bats. However, there are a lot of filoviruses, and not all of them are found in these bats specifically. For example, Bombali virus has been found in these guys, as well as in Mops condylarus, and Yavu virus was found in Miniopterus bats in Europe. And then from there, it gets weirder. We've got the Stria viruses found in saltwater fish. Those guys have so many different names. I'm not going to try to tell you which one that is. But these guys were found in saltwater fish. We've got some found in snakes, found in freshwater perch in Europe. And finally, the thamnoviruses found in, I believe, the greenfin horse-faced filefish, which sounds like a playground insult, but is in fact a source of filovirus sequences. So there's a lot more of a broad and rich field to play around in than just what we consider sort of the flagship or model viruses of a family, which obviously for the filoviruses is usually Ebola virus. So to kind of get a better idea of characterizing these other viruses, generally what we would hope for from these bats or fish or snakes is the opportunity to just directly get a viral isolate. But unfortunately, many of these viruses have not actually been found in an isolated form, and many have never been found in, for example, primates or humans. Generally, that's a good thing. But it does make it a little bit harder to understand what's going on with them and get a better idea of their molecular biology. Generally, what we get instead are sequences that, based on structure, appear to be a complete filovirus genome, or even more frequently, an incomplete filovirus genome with missing coding or non-coding sequences, especially near the ends. When what we get is a complete sequence, one of the options is to construct a recombinant virus out of that. And this isn't my work to get into, so I won't be delving into this today. But we've done some interesting work previously using this approach to study Yavu virus prior to the first isolation of infectious Yavu virus from bats. In addition, my lab mate Stephen, who recently defended, has come up with a really cool solution for the problem of degraded 5' ends in filovirus sequences. So I encourage you to go, that's my plug for his paper. So we tend to take an approach to understanding filovirus molecular biology that's a lot more kind of a nitty-gritty cell-based level than the kind of elegant ecological scope things that we've covered today. Starting with the familiar filoviruses, I'm just going to give kind of an overview of what's going on in the cell. Filoviruses enter through a larger macropinocytosis type mechanism, come in in the endosomal network. And what we consider to be the entry receptor, NPC1, for filoviruses is really what's necessary for fusion of the filovirus membrane with the internal membrane here to allow escape into the cytoplasm. And so generally, when a filovirus sequence is discovered, the way that people approach finding out is this likely to infect humans, for example, is to take that glycoprotein coding sequence, which is often intact, and generate a pseudotyped viral particle, usually VSV, and see if that can get into a cell of the species you're interested in. This is a great way to start to find out if a virus can get in, but viruses have to do a lot more than that in the cell, such as transcribe their RNA, replicate their genome, evade host immune responses, assemble new progeny viral particles, and escape. Those things also involve interaction with host proteins, and so we really want to know, how can viruses act once they're in the cell, and what do their actions within the cell look like? MIDI genomes are kind of the bread and butter of our lab, so I'm going to just kind of review what these systems are. we take the viral genome, remove some or all of the coding sequences, and replace that with a reporter flanked by authentic viral sequences. This we insert into a plasmid, and in combination with support proteins, encoding viral proteins for transcription or replication, we can transfect into cells together, allowing for transcription of a viral mini genome and production of viral proteins, which can then accept that mini genome template as if it were a real viral genome, transcribe it, and produce both reporter gene signal and more progeny genomes. Since we've done a lot of this sort of work, I'm going to start with this to kind of discuss the project that I've been largely working on, one specific interesting filovirus protein, VP30. VP30 is particularly interesting because it's specific to the filoviruses. It's a phosphoprotein, and we know that it serves some transcriptional functions. And for Ebola virus, we even know where in the genome it acts. Interestingly, replication of the viral genome by the polymerase and its associated proteins is VP30 independent and only requires VP35 and NP to work with the polymerase. However, for Ebola virus, we know that VP30 acts. Is my mouse visible? Awesome, thank you. Acts here at this hairpin structure formed between the transcription start site of the first gene and the downstream sequences. This hairpin structure actually seems to dictate whether transcription can be activated at the start of the genome or not for Ebola virus. And this is particularly important because transcription starts at the 3' end of the genome and cannot initiate internally. So this transcriptional failure, if it occurs at the very first gene, this makes an insurmountable problem for Ebola virus. Without transcriptional activation at NP, we end up with no NP mRNA or protein produced, and the polymerase complex also cannot move forward. So as a result, nothing downstream in the genome gets produced either. However, not all filoviruses are Ebola virus, like I mentioned before. This part of the process happens just fine, for example, for Marburg virus, but something else fails in the transcriptional process if we attempt viral rescue without VP30. So this makes Marburg virus a perfect platform to investigate additional VP30 functions, which is kind of my pitch. Specifically, I'm interested in internal transcription events, which we can't observe in Ebola virus because they occur downstream of that first gene. That smooth linear motion I showed on the previous slide was a lie, by the way. Stopping and starting at gene borders is how the polymerase complex makes these individual monocistronic mRNAs for each of the genes. And because each of those gene borders requires reinitiation of transcription and can fail, we end up with a gradient in quantity of monocistronic mRNAs. Because of this reinitiation, we modified our mini genome system to study relative gene expression across these borders. Again, based on that mini genome system, we then made a bicistronic version using an authentic viral gene border in between two different reporters, which we want to know if VP30 acts in between. These produce varying quantities of mRNAs, and if VP30 is necessary for reinitiation, we should expect to see a proportionally greater drop in the second gene relative to the first in the absence of VP30. So using this system, we're presenting these results here as a ratio from the second gene to the first gene. And if VP30 is required for transcriptional reinitiation across this border, we expect to see a drop in that value in the absence of VP30. Which, using the NP to VP35 gene border, the first one in the genome, we really don't see. However, as we were working on this, another group published some findings indicating that VP30 was actually required for transcription initiation at the glycoprotein gene, which is further down in the genome between VP40 and GP. So I went back, removed and replaced that NP to VP35 border with the VP40 to GP border of Marburg virus and repeated this experiment. And interestingly, I did not find a significant drop in this relative gene expression ratio. That was kind of a surprise. But because I'm also curious about other maybe more obscure filoviruses. I also tried this with Dianlovirus mungla, which I apologize, I'm probably not pronouncing quite right, using the VP40 to GP border of that virus. Again, we really didn't see much of a difference. This discrepancy with published findings is interesting, but it raises more questions than it answers. And to probe VP30 transcriptional events in a more authentic setting, since these experiments have all been performed in a mini genome, we moved into systems of more complexity and added back in some of those viral moving parts. We've used some systems of intermediate complexity that I don't have time to cover here to find that swapping out these gene borders or changing the structure of the hairpins there did not change the need of VP30 to propagate virus-like particles. We jump from there to recombinant virus generation, and I'll explain why and how. First, the why. We wanted to use a recombinant viral particle approach, a recombinant virus approach to study VP30 because the previous systems are all transfection based. And I think we can all agree that although as convenient as it is to be able to remove just one plasmid and have a protein not present in your system, it's really not the same thing as removing it from an actual viral infection. And as we were puzzling over how to best remove VP30 from a viral infection, because siRNA is just not very temporally controllable and frankly kind of cumbersome and messy, especially for BSL-4 experiments, our colleague, our collaborator Sean Lyons, who works on cellular biology, pitched to us this Degron system he had for targeted degradation of cellular proteins. And I decided I want to do that with viral proteins. The way the system works is you tag your protein of interest, in this case VP30, with a peptide, a Degron, that's recognized by a soluble mediator, which, when added into the system, facilitates an interaction between this protein of interest and cellular F-box proteins, which form an SCF complex and recruit a ubiquitin ligase to polyubiquitinate your protein of interest and target it for proteasomal degradation. This is particularly cool because it's temporally controllable and a lot more immediate and finely tunable than siRNA transfection, and you can do it after infection starts. This is particularly interesting because then we can isolate different parts of the viral infection cycle. So to rescue infectious virus from the with this tag from cDNA, we follow a very similar workflow to a mini genome, except in the BSL-4. Instead of removing the viral genes, we put the entire thing into the entire viral genome into a plasmid and transfect it into cells with plasmids encoding NP, VP30, VP35, and L. These proteins then get produced and encapsulate the viral genome, produce viral proteins, and eventually combine with that genome to produce viral particles. It's worth noting that not all viral proteins necessarily tolerate being tagged. Some of them simply will generate a non-viable virus. So the first part of this proof of concept that I've worked on is finding out whether we can even rescue the virus and whether it'll tolerate a tag on VP30. I'm delighted to announce, as you can see in this Western blot, that we did get viable virus and that the tagged VP30 protein down here is visible under non-degradation conditions. Even more exciting was that after a full day of infection, when I added in this soluble mediator auxin for I believe for this experiment, I did 4 hours, we get what I believe was quantified as about 85% loss of protein on the first try, which was really, really exciting because this is showing that even once the virus has a whole day of head start, we can degrade this out of the system. And it still actually works. Apparently the cellular degradation machinery is robust enough to handle this. So I'm still kind of fine-tuning the timing of these experiments and ways to get even more complete degradation than this, but I also was really excited to see where this would lead transcriptionally, and so I started off with a pilot experiment using RT-PCR to just assess viral transcripts. I infected cells and treated them with this degradation compound at the time of infection or later on. and found that I got some kind of noisy signal because the even with the wild type virus, because the cells apparently did not like having that much DMSO in them. Who knew? But in comparison, almost no viral transcripts were observable relative to the vehicle control. In the degradation tagged virus, when I induce degradation at the time of infection, these early time points are clearly a very important part for VP30, a very important part of the viral infection cycle for VP30. And also interestingly, I didn't see as stark of a drop across any of the particular gene borders as we were expecting to based on those published results I had mentioned. So my working model, and this again is quite preliminary, this is a little bit of a noisy system that we're still setting up, but I'm very excited about it. And this kind of works with my working model here, which is rather than one specific gene border dictating the function of VP30 in transcribing across the entire Marburg virus genome, that more likely instead, transcriptional reinitiation is a process that requires VP30 to be efficient across all gene borders. A cumulative loss of efficiency across the genome could be disastrous for viral replication because you don't get enough polymerase produced. And I think that presumably this transcriptional Filominomenon is not specific to Marburg virus, but it's previously been unobservable for Ebola virus due to that transcription activation defect I described before. So the next steps in this that I'm looking forward to are to use these viral reverse genetic systems to find out what's going on here by running an RNA-seq experiment, which I'm currently collecting samples for, to actually get a genome coverage map across the entire viral genome to better see if there's specific regions targeted in transcriptional defects in the absence of VP30, or if we see, like I predict here, this global exacerbation of an existing transcriptional gradient. And for the future, maybe more filoviruses, I have a system that I don't have time to discuss here, but a way to perhaps liberate Ebola virus from that VP30 dependence of transcriptional activation at the NP hairpin. And I apologize that ran a little bit long, but I'd like to give a shout out to my lab. And if there's time for any questions, I'd be happy to see them. If not, you have to look at my cat.